transaminase enzymes involve alpha-ketoglutarate and glutamate as a pair. Otherwise, you get a whole bunch of amino acids, and there are about 11 of them that participate in this reaction, but they all donate their nitrogen to alpha-ketoglutarate. So alpha-ketoglutarate accepts nitrogen from 11 amino acids or so. It's a main part of the funneling exercise. So, go to one thing that's usually tested somewhere, is all these transaminase reactions require pyridoxal phosphate as a cofactor. I think Dr. Graham's giving a lecture tomorrow, which should be very useful to you because it's high yield stuff. Vitamins, minerals, cofactors. Be there. All right, so you got a lot of excess nitrogen being funneled to alpha-ketoglutarate. Now, the other thing that happens is there are a number of amino acids that lose their nitrogen, not by transamination, but by deamination. That's this slide here. And it shows like histidine and threonine and serine. They all produce ammonia. Ammonia comes from a variety of other sources too. Significantly, bacteria in the gut break down things like urea. So, to ammonia. All right, so what you've got is a whole bunch of nitrogen that's been funneled into glutamate, and then you got free ammonia. Well, from glutamate, you react with oxaloacetate in a transamination reaction to produce aspartate. So now you've got the two key molecules that donate nitrogen into the urea cycle. So if you were to be asked, what are the key sources of nitrogen that are donated into the urea cycle, I would say aspartate and ammonia. All right? So here's the ammonia. It comes from a variety of places. The aspartate through a whole bunch of transaminations. Urea goes from the liver to the blood to the kidney and is excreted in the urine. It's our main excretory product of nitrogen. A key reaction that controls the amount of ammonia in the blood is glutamate dehydrogenase. What this does, this is not a transamination reaction. Here you have glutamate reacts with NAD or NADP. This gets oxidized to a carbonyl group and ammonia is released, and this reaction is freely reversible. When the level of ammonia in the cell gets too high, this reaction moves right to left just by mass action, the Chatelier's principle. You get rid of that nasty stuff. Remember, ammonia in high concentrations is extremely toxic. It's neurotoxic. Other key reactions that control the level of ammonia is a glutamine synthetase reaction, where glutamate combines with ammonia to form an amide bond. This is on the carboxyl side chain of glutamate. That gives you glutamine. So glutamine is also an important carrier of nitrogen through the blood. To go back the other way, glutamine is hydrolyzed by the enzyme glutaminase, or by water, catalyzed by glutaminase back to glutamate and ammonia. So glutamine and alanine are the main carriers of nitrogen through the blood. Alanine's fate is usually to be picked up by the liver, and where the carbon skeleton is converted to glucose. Think of the carbon skeleton of alanine as pyruvate. Think of the carbon skeleton of alanine as pyruvate. So, alanine goes to the liver, is carbon skeleton, pyruvate goes to glucose, and the nitrogens are released and wind up being excreted as urea. Glutamine has a number of other fates because it has a number of important uses. So let's go through some of these bullet points, make sure I didn't forget anything. So ammonia is toxic. That's sort of the theme of nitrogen excretion, is the toxicity of ammonia. And we go through all this rigmarole to keep the level of free ammonia down and make sure that ammonia is being transported around in non-toxic forms such as amino acids and urea. Urea is the main non-toxic excretory product. And it goes out in the urine. The urea cycle, nitrogen enters as ammonia and the nitrogen of aspartate, and it leaves us urea. The first step here, this is kind of the cycle, but the first step is not technically part of the cycle, but it's ammonia plus CO2 gives you carbamyl phosphate. Key enzyme, carbamyl phosphate synthetase. I would know that one well. Cycle is split between the matrix and the cytosol. Now notice here's aspartate coming in the cytosol. So aspartate comes in and fumarate leaves. So aspartate has this ammonia. It leaves its ammonia behind, and then but the 4 carbons, the skeleton, take off as fumarate. So fumarate is a product of the cycle, and something must be done with it. You can't just keep accumulating fumarate every time you're on the cycle. So fumarate, that's another area to know, is what becomes a fumarate. Fumarate can either go to glucose in a fasting state or actually recycle back to aspartate so that it doesn't build up. When it does recycle back, you have what's known as the Krebs bicycle. So I would know this urea cycle real well. This is a very popular, well-tested cycle. And you know there's so many disorders of the urea cycle. I think I told you during the lecture, you can expect a question where you're either given an enzyme that's defective and you have to select what are the most likely pattern of metabolites that are too high or too low. Conversely, a patient is brought in, what's the usual thing? Lethargic, vomiting, hyperventilating, God knows what. And then you're given a bunch of lab tests. There's high ammonia, there's low this, there's high this, there's low that. You've got to figure out what enzyme is defective in the site. So it starts with carbamyl phosphate and any of these enzymes here. So I gave you an assignment in Marks. There's at the end of the chapter on this. There's a pretty good question that kind of leads you through this. If you can answer that question, the answers are in the back of the book anyway. You should be all right. But make sure that you're good at that. Then there's the use of benzoate and phenylbutyrate to treat the urea cycle, to treat inborn errors. Basic idea there is these are drugs. So many drugs are excreted as derivatives. Now, we just got through the MCAD thing. We find that a lot of these MCAD waste products are excreted as glycine derivatives or carnitine derivatives. Benzoate and phenylbutyrate are excreted as other amino acid derivatives. I can't remember which ones they are, to tell you the truth. But the basic idea is that you're losing nitrogen through the urine when you do this. And so these derivatives of benzoate and phenylbutyrate serve the role of urea, of eliminating waste nitrogen. Okay. So let's move on to the lecture on the